Anytime she goes away.
So hi everyone, I'm here to talk to you about the coolest protein on earth. Well, it's a rhodopsin. You all have rhodopsins in your eyes right now, and you're using rhodopsins to look at me. Yet few of us know how they actually work, right? So what few of us even know is that microorganisms also have rhodopsins. Algae have them, bacteria have them, viruses have them. And they use it to do different things in reaction to light. They can swim towards light, they can swim away from light, they can even turn on kinases and turn on transcription in reaction to light. They can do many different things. And although humans and microbes are very different, the trick is always the same. So what is a rhodopsin? A rhodopsin is a single protein, okay? And it's made with 7 transmembrane helices, which are packed this way into a 7 transmembranal protein. Here, I'm going to fix it, sorry. It worked much better at home. Okay, so it's a 7 transmembrane protein which is packed this way. And the rhodopsin itself, it's nothing without its power core. So the power core of the rhodopsin is vitamin A, or retinal, which I also have in my pocket. So retinal looks like this. It's a simple molecule, it's a chromophore. It has a ring and a tail. And when photons hit this molecule, what actually happens is that the tail bends. So a single photon hits this molecule and the tail bends and then unbends. So if this happens a lot, what happens to the tail, it goes. And so it's actually like a machine that is moving in reaction to light. So what happens when you put this molecule inside the rhodopsin? It goes inside the rhodopsin, and now there are functional mechanisms. Because when light hits this molecule, it's also trying to move inside the protein. Because the protein holds it from one side and from the other side, the movement of the retinal actually moves the helices as opposed to each other. And this movement is what creates the machinery of this protein. The movement can create pumping of protons outside of the cell. It can create movement of different ions across the membrane of the organism. And in the case of a channel rhodopsin, they can actually open up a specific ion channel which will move ions very rapidly across the membrane. And this channel rhodopsin, which was discovered, made a huge difference because somebody realized that you can take this rhodopsin and put it inside any neuron that you have, and now this neuron will react to light. This was the birth of the field called optogenetics. So in the field of optogenetics, you're using channel rhodopsins to stimulate single neurons or single populations of neurons in reaction to light. And you can study disease and you can do research on single neuron level. And I'm pretty sure that there will be a Nobel Prize very soon on this subject. So this was the motivation for me why we should look for more rhodopsins. I mean, when you have a toolkit, different rhodopsins can react to different wavelengths and they can carry out different functions. So searching for new ones is what motivated me to search for them. And we used a really cool, simple trick. When you express rhodopsins inside E. coli, it's just a protein. It's yellow. It doesn't actually have color. But when you place the retinal inside the molecule, not the retinal molecule, inside the protein bends the retinal just a little bit, and the molecule, which is actually yellow, together with the rhodopsin, has a different color now. So the E. coli itself will turn purple, red, or orange. So we just used this cool, simple trick. We took random DNA from the environment and put it inside E. coli. And when we gave it retinal, the E. coli itself turned red or purple or orange. And we were hoping to find new rhodopsins, and we actually did. We found a whole new family of different rhodopsins never been seen before, which we named Heliorhodopsins. And the reason why they were different is because they had no similarity by sequence to the human rhodopsins or to the microbial rhodopsins, yet they were a huge family, also existent in all kinds of microbes, algae, and bacteria and viruses. And I mean, what was also different about this rhodopsin is that it was the first rhodopsin ever to be completely inverted in the membrane. It was a 180 degrees inversion, so the inside was outside and the outside was inside. And this is what happens in this family of rhodopsins always. So we have a new family. We found it in 2018, and almost 2 years later, 3 crystal structures later, 41 citations to the original Nature paper, and we still don't know what does it do. It's a light switch. We know it's a light switch. We know what wavelength switches it on, but still we don't know what it does. I mean, it's just a light switch on a blank wall. We just don't know what it does. So when you want to figure out what it does, you need to go back to nature, to the organisms which express it, to figure out what it does. And what I'm putting my money on is on this story. We have Alina Huxley algae, which are causing these huge blooms, which you can actually see from space. And these blooms are killed off by a virus called the Alina Huxley virus. And all the Alina Huxley viruses always have Heliorhodopsins on them. And we don't know why. Why would a virus need a light-sensing protein? I mean, it's not motile. It cannot swim away from light. It cannot swim towards light. What does it do, right? And viruses don't keep things just for no reason. It's probably a massive, there's a story behind here. It's crucial to the infection cycle. And these proteins are not just in the DNA of the virus. They're actually expressed. The group of Asaf Vardi showed that they're expressed on the protein level on the viruses. This is also unpublished. Don't scoop us. And so when I want to figure out why a virus like this wants to infect by using such a protein, I turn to the world of criminology. Okay? So you want to know the motivation, you should think like the killer. So let's say that all of you are green photosynthetic algae. Okay? You see the green? And so when do you want to infect all these nice photosynthetic algae? Let's say that Oded and Shai are the viruses. They want to infect this algae during the light, because in the light, the photosynthetic organism does photosynthesis. It has a much better metabolic state. So this is one motivation. And secondly, you want to infect when you have a lot of green around you, right? Because you want your progeny to have also someone to infect. And what we think is that the Heliorhodopsin is the way that the virus decides to infect, specifically during the light and specifically when it sees green light around it. Because when we see that when we express these Heliorhodopsins inside E. coli, most of them have absorption and green-blue wavelengths. So what I'm suggesting is that perhaps we will soon have the answer whether viruses are alive or dead. I mean, if viruses can actually sense light, it will be a definite answer. If it makes a decision, if it turns on some kind of transcription, if it does something with the lipids even before it touches the host, it's a definitive answer that the viruses are actually alive. Thank you.